through the first part of transcription, which only requires the one enzyme, which is really more of an enzyme complex. It's got its own little helical built in to split the DNA in the front. It's got parts that'll add mRNA bases, complementary bases. Remember that there's U's instead of T's, but everything else is the same. And then close it back up in the back so you wind up with the DNA re-zipped like it was, and then a primary mRNA transcript. Well, the thing is that primary mRNA transcript has some bases in it that aren't supposed to be there. Stuff that needs to get edited out before the mRNA gets to leave the nucleus. And that process is called RNA editing. RNA editing is done by another big old enzyme complex you don't need to know the name of. It's kind of a fun name, though. It's called the spliceosome. And there's a pair of terms here that, for me, always seems backwards. That's sort of how I remember which is which. The junk parts of the mRNA are called introns, and the parts that are necessary are called exons. If just remembering that it's backwards from what you think it is isn't enough, one other little trick is the introns aren't good enough to leave the nucleus, so they have to stay in, and the exons get to exit the nucleus as code. So working with our little strand of mRNA, and sort of just for example's sake and very simplified, let's say that these are the parts that are important, and this is the part that's not. And once you've removed all the parts that need to be removed, then the edited mRNA is ready. Now, there's a couple other modifications you make. You put some adenines on the front. They call that the poly A. Or no, you put the adenines on the back. They call it a poly A tail. And they put a methyl group on the front called the methyl cap. You don't have to know that. We'll stick with the basic editing. And so edited and spliced back together, here is your final mRNA transcript. Keep in mind, this is an example. In reality, this thing is much longer. There are many more segments that are introns that get snipped out. And then, no, there's no way you're going to be able to predict what comes out and what stays. And so at this point, the mRNA is ready. It's edited. It's ready to leave the nucleus, head into the cytoplasm, where this kind of multi-part dance is about to start, where proteins get built. It's fairly complex and is going to require a bunch of drawing from you. What's nice is that we don't have to do that. We get to cheat. Because we know the code. Just from the mRNA, we can actually tell which amino acids are going to get coded for in that protein. We don't have to go through all the machinery and the 3 kinds of RNA that are going to be involved. We're going to do that tomorrow. We'll show you all that stuff. But today we're going to jump ahead and just show you the code to get from mRNA straight to amino acids. Are there still prizes in cereal boxes? Do they still do that? And I love that. I know that, like, by the time I even got to college, they had started not burying the prizes in the actual cereal. I think probably for hygiene's sake, because I can remember many times being, armpit deep in the Cheerios trying to fish out the thing that was the prize. Anyway, I don't know if they do that anymore or not, but one of the cool things you could get sometimes was a little secret decoder wheel. You know what I'm talking about? Like, it was just a simple little substitution code. Like, this is a very old version before my time, but where'd it go? That's like 1935, right? But the principle's the same. You turn the wheel so that each letter gets a number or each letter gets a different letter to substitution code. And you can write something to your buddy if you both have the same wheel, right? And your buddy can decode it and it says, your buddy's stinky or whatever. Anyway, basically, you're about to get your own secret decoder wheel. Because the code is always the same. And it's always read 3 mRNA bases at a time. Those sets of 3 bases are called codons. And I bet you can figure out how to use it without my help. At least some of you can. It's a fairly intuitive tool. Keep in mind, this is not the only tool that does this. There are many ways to kind of make keys for the code. This one in particular, the thing you absolutely have to remember is that this wheel is made for mRNA only. You don't put DNA into this, and you don't, once we get into translating tRNA, you don't use it with tRNA either. There are wheels that are built for tRNA. This is not that. This one is so that you can cheat and skip ahead. You don't have to deal with tRNA. Can you tell how you're supposed to do it? Somebody want to tell me what they think the first amino acid coded for by these 3 letters might be? Amanda?
Methionine.
Methionine is how you say it, and you're absolutely right. Everybody see how she got it? Okay, let me make it real simple. You see the really big letters in the middle? That's where you start. And then you work your way outward through the 3 letters in the codon. So A was the first letter, which restricts you now to the, let's call it the southwest quadrant of the wheel. Then U in the middle row here, right? We get to U, and that's somewhere between like 6 and 7 o'clock if this was a clock. And then G restricts you to just the one amino acid, which is methionine. Go ahead and use the wheel. You can write down methionine, MET, or you know, first 3 letters of the amino acid is fairly common abbreviation. Go ahead and write the abbreviations of the amino acid that this whole thing would code for. Easy? Got it? What are we looking at? Let's see. For the second set, which is CUC, what is the amino acid that codes for? Sam, what do you think? Leucine, exactly. Notice with leucine, and you can look around and see that it's the case with more than one of these. What would happen if you changed that 3rd letter in CUC to something else? What would it code for if you changed it to a G? It kind of doesn't matter what's in that 3rd spot for amino acids like leucine, like valine over on the left-hand side, and alanine. That 3rd base can be kind of a flexible thing. In fact, sometimes they call it a wobble base. It kind of doesn't matter if you were to get a mutation there. And you'll notice there's more than 20 slots around this thing. There's only 20 amino acids in 120 slots, so some of them appear more than once. What else do you notice that's kind of weird? There should be 2 spots there.
The 2 stops.
There's actually 3 different codons that instead of an amino acid name there, it says stop. What do you suppose that's all about?
Where the RNA and the polymerize stop coding.
You got to have some signal there to know when your gene is done transcribing and translating. That would be it right there. That's how the whole assembly that's building this protein knows to stop translation, snip off the long chain of amino acids, and let it fold up into a functional protein. That's the stop signal, okay? So that's the idea. I'm going to give you a bunch of practice doing that on a real protein here in a minute. One more thing I'd like to mention. Oh, wait, I forgot. What's the third one? Matt, what's the third amino acid, AAG?
Oh, lysine.
Lysine. So methionine, leucine, lysine. And again, the first three letters of most of these is a perfectly good abbreviation. All right. One last thing about RNA editing. One of the really interesting things about the Human Genome Project, when we sequenced the entire human genome and started to connect that sequence to actual genes for actual protein, is that we knew that the number of proteins in a human is way, way, way bigger than the number of genes we actually found in the genome. There's only about 25,000 genes that we found. And yet the number of proteins that a human can make is much, much, much larger than that. And there's a couple of different explanations that scientists are sort of between. But one of the things we know for sure is that some of that discrepancy, some of the difference between how many genes there are and how many proteins there are, has to do with RNA editing. Because, of course, small changes in the sequences of amino acids in a protein can change the function of that protein drastically, can make it a completely different protein. And so one way to get some flexibility to be able to use one gene to code for more than one protein would certainly be to edit the transcripts differently, right? Different ways of editing the mRNA, that'll change the sequence. That'll make it a totally different protein. And so that is one of the ways that we are relatively certain that multiple proteins could be made from one gene. All right, I want to make extra sure that you all get how to use the wheel. And so I'm going to give you a real protein to work on. It's insulin. However, human insulin is a little too long. So we're using an animal that has a simpler version. We're using a cow. The cow version of insulin is pretty short, and so we can afford the time to take the, you've already been given the mRNA. You're going to turn the mRNA into the amino acids. And then I am going to have you backwards transcribe, reverse transcribe the mRNA back into DNA, just so I'm double sure you're good at that whole complementary base pair. Going to need the wheel for sure. That wheel is going to be your constant companion for the next week or so. Please understand, I'll never, ever make you memorize the wheel. You'll always have the wheel if you need to. For example, if there were a quiz and you wanted to make you do the translation, give you the wheel on the board. All right, take a look. I think it'll be fairly evident, except that the mRNA is very small. So I went ahead and divided up into threes for you with the little lines. And so you go 3 by 3 along the mRNA and just write the first 3 letters in the little numbered bubbles, the first 3 letters of the amino acid that mRNA would code for according to the wheel. The first 4 are done for you so that you know you're doing this right. That'll help. Make sure you pay attention to those numbers. Then you've got 2 questions to answer. And then, yes, you do need to do the thing at the bottom where it says DNA sequence from which that RNA was transcribed. So help me out for those first 3 letters, U, and the mRNA. What would be the DNA that came from? A would be the first 3 letters of that. Go ahead and write that down just so you understand what you're doing. Not a terrible idea if you're not quite there on the complementary bases. Not the worst idea when you do that backwards transcription to go, okay, I know there's A's. Sorry, I'll do it up here. Make yourself a little key that, like, okay, if there's if I got mRNA and I'm going to DNA, if there's an A here, I know it was a T here. There's a U here, I know it was an A, C, G, G, C. And that way you kind of have it in front of you. Not always going to be able to do that, but not a bad idea for training wheels. All right, I'm going to give you the time to work.